Okay, hi everybody. I think we've got most of the people we're expecting. I think we've got all the students on the call, which is the most important thing. Um, my name is Sarah Cross. I am the director of the master's program in Ecology, Evolution, and Conservation Biology at Columbia University, and we are here today to celebrate this year's graduating class. So I'd like to welcome everyone to our 2020 Research Showcase. Obviously, under normal conditions, we'd be holding this event live and in person on campus in the days around the normal commencement celebrations at Columbia. But of course, this year is different than any other in recent history. And it brings us immense challenges, but also opportunities. So today's call on Zoom allows our graduating students to share their work with families and friends. I can see a bunch of you out there, and cats and dogs, and maybe some other pets too. And you are literally bringing us into your houses. And this is something in person we would not obviously be able to do. So we're really excited to have so many of our graduates' loved ones on the call today. And I think you're going to be really amazed and impressed at the work that they have done. So to kick us off, I would like to introduce Professor Shahid Naeem, who is our department chair and who is going to say a few words. So Shahid, I think you should be able to take over.
Unmute. Can you hear me?
Yes.
All right. So thanks, especially for the challenge to say something in just a few words, because I'm famous for not being able to do that, but I'll try to keep to my time. That's hard to do when it comes to the graduate students in our program, the master's students especially. But if you think about it, all of the students we're seeing finishing up today and the students who are continuing, and in fact the students are coming soon, they had only 2 years, just 2 years, to sort of learn all of the natural sciences, physics, chemistry, math in one form or another, as well as ecology, evolution, and environmental biology. But they had to learn, modeling, statistics, mastering computationally intensive analytical tools like GIS and R and so forth. So we're going to be celebrating the outcomes of those sort of 2-year endeavors, and some of them are continuing on. And I think what you'll be impressed about is how extraordinary, wide-ranging the research has been and the topics for such a small program. So, I think in this graduating cohort, we will be hearing about evolutionary primatology, conservation biology, disease ecology, landscape ecology, community ecology, my field, biogeochemistry. And people have worked as far away as Kenya, India, China, the Amazon, and some people right here in New York, which may be more challenging than some of those other places. Working on urban ecosystems, rainforests, montane habitats, wetlands, and they've worked on everything from domestic cats, one of my favorite species, to wild cats like ocelots and tigers, which I've always liked as well. Studying dominance and aggression in blue monkeys, olfactory communication, nitrogen fixation in trees, and on it goes. And if we were to cover the students before and the students coming, you'd see that there's almost no topic that hasn't been picked up by our master's students. And so if we were, I imagine if we try to sort of put pins on a map, even with a small cohort, but for our master's program in general, almost the entire world would be covered. Maybe not the Mariana Trenches at the bottom of the ocean, but most everything else has been covered. And you'd find that if you could find some way to sort of map the disciplines, ecology, evolution, and environmental biology, all the fields and all the subfields would pretty much be covered by this small department. And if we were to map the ways that the science of the living world could actually be leveraged to make the world a better place, I think you'll see that our RMAs have done it. I've been here for some 17 years, I guess, and the program was still here before even I started. But I think what's amazing is where our students have gone on in the environmental arena, whether it's working in NGOs or whether it's working in private institutions. Some have become teachers. One has become a wildlife photographer, and researchers. Many have gone on to PhDs because they just couldn't, get enough of the challenge and the torture of doing rigorous scientific research. And I think that we shouldn't lose sight of the fact that what makes it possible is that our department is actually, I find, although small, it's the smallest of the arts and sciences department at Columbia University. But given its intellectual domain, I've argued, and I've said this in front of the vice president, the president, the chairs of the university, of different departments of the university, that we have the largest intellectual domain, larger than cosmology, larger than physics, larger than anything in the humanities. And yet we're able to cover them really quite well. I think we do that because we have an incredible set of faculty, adjuncts, postdocs, technicians, and students from undergraduates to PhDs, in our master's students, as well as an amazing staff. We have a small but incredibly amazing staff that makes it all work. So to me, it's just one of the most exciting times of the year, and saddest, of course, because we all are going to leave. These are trying circumstances, and yet I think people have risen to the challenge. I'm looking forward to seeing this event, even though I'd much rather be there and opening a bottle of champagne with all of you. But it is nice to actually to make sure that we were able to celebrate this really important event. So thanks very much for making the world a better place, for rising to all of the challenges that we put you through. I'm not sure I could have survived them. And for continuing on to carry on the work of E3B and what we built. I think the other thing that I like to keep in mind is that what you've contributed is not just to your careers, but also to E3B. Every year, E3B becomes a stronger place and is capable of doing, here's the domestic cat, doing more amazing work thanks to you. So your legacy will also live on as well. So thanks, everyone. Congratulations to you. I think I may have stayed in my 5 minutes, but I'm not sure. So thanks, everyone.
I think that was spot on. Thank you, Shahid. So I wanted to point out that I think that the class of 2020 are just incredible. They're a tight-knit, funny, and extremely hardworking group. They've worked on a diverse range of projects, as you're going to see in a few minutes, and I've just been so amazed by how much they've taught and learned from each other over the last 2 years. In ecology, we commonly use the term resilience. A resilient ecosystem is one that can withstand disturbance and return to normal patterns and functioning afterwards. And I think the students we'll hear from today are perhaps one of the most resilient groups I've had the pleasure of working alongside, although I think next year's class might top them. But they came into the department with a totally green program director who didn't know anything about the program at the time, really. And shortly afterwards, we had a major change in our administrative team. Then they had to face the sudden loss of a faculty member in our department. And they've had their own challenges along the way, but their personal and collective determination and resilience has shown through. In the final months of their masters, at a time when all students are normally under extreme pressure as they wrestle with data analysis and turning an immense amount of reading and work that they've done into a cohesive final product, this group has faced a global pandemic. And so they've scattered across the country and they've had their committee meetings through computer screens, just like this one. They're working on couches in their childhood bedrooms to finish up this big piece of work. And I'm not surprised that they've all made great progress in this, because they are students that are ultimately robust. They're students that have somehow remained funny and enthusiastic through all of this, and students that are ultimately resilient. You family members out there, I'm so glad to see you today. I'm so glad to meet you finally, because I've heard a lot about you over the last couple of years. You're a huge part of the connections within our academic ecosystem, and you're a core of what makes us so resilient. So I know you may not always know exactly what these students are talking about when they get all excited about their work, but having you there to smile and nod and give them encouragement is what makes them so robust. So Rachel, Hannah, Laura, Alexa, Michael, Emily, Jared, we are so proud of everything you've done in the last 2 years, and collectively as a department, we want to say congratulations to you. And now I will stop talking because I would love to introduce our first speaker today. So first up, we have Rachel Donabedian. She is going to be talking to us. So Rachel, go ahead and start sharing your screen. She will be talking about mechanisms of dominance rank acquisition in a primate with low aggression and coalition rates. Her committee were Marina Cord, Scott Siller, and Eleni Nikotopoulos at the New York Institute of Technology. And her peers have told me that they think of Rachel as the kind of core of this group, as one of the key members. And I'm sorry, one of my children is screaming in the background, so I will mute and let Rachel take it away.
I did my thesis on dominance rank acquisition in blue monkeys. And so I know many of you are probably wondering what dominance rank acquisition even means. So in many species with social hierarchies, juveniles, as they grow up, will often attain a social rank that is similar to their mothers. This is called maternal rank inheritance. However, in species with maternal rank inheritance, infants are typically born low ranking. So they start at the bottom of the hierarchy, which means that as they grow up, they have to increase their rank to reach their mother's position, which begs the question of how do juveniles actually go about this process of increasing their social rank? And in other species with maternal rank inheritance, which include many species of monkeys and also hyenas, prior research has shown two main ways this may be done. One is by targeting individuals who rank below their mother in aggressive interactions. So maybe they'll specifically direct aggression to the individuals they would be expected to outrank in adulthood as they get older. The second way they may go about this is by receiving support from their kin and relatives in coalitions. So perhaps a juvenile will challenge an individual who she might not be able to beat on her own, but then mom or another family member will come and back her up, allowing her to win an interaction she otherwise would not. And the reason that to look at this in blue monkeys, rank acquisition, is because the other species where rank acquisition has been studied are fairly aggressive species, and they also form coalitions frequently. Blue monkeys, on the other hand, have fairly low aggression rates, and coalitions are pretty infrequent. So I wanted to know if rank acquisition occurs differently in a species that still has matrilineal rank inheritance, but overall shows some different patterns of social behavior. And so to answer this question, I looked at a data set that had over 10 years of dominance interactions between blue monkeys living, a wild population living in Kenya. I then used an algorithm called ELO ratings to track the ranks of juveniles from birth until they reach their mother's rank, and then look at if there were other behavioral changes that accompanied this process of their rank increasing. And so for my first question, looking at if juveniles increasingly target individuals who their mom outranks, short answer is that I generally did not find evidence that juveniles were targeting individuals from lower ranking families as they aged. If that were the case, we'd expect to see an interaction between age and whether or not they were challenging an individual their mother outranked, which is, however, what we saw is actually what's depicted here. The dark blue line shows interactions with individuals their mother outranks. The light blue line shows interactions with individuals their mother does not outrank. So those are the individuals they are not expected to outrank in adulthood. We did find was that at all ages, juveniles directed more aggression to individuals their mother outranked, even starting in infancy. We also found that juveniles increased their aggression towards everyone as they got older. However, they were not specifically increasing aggression towards those their mother outranked. And then for my second question of looking at how important coalitions are to the process of rank acquisition in blue monkeys, to sort of try to get at this problem, I looked at the social outcomes of individuals whose moms weren't present in the group, so orphans, because moms would presumably be one of their most important coalitionary partners. So first, I wanted to see if juveniles could even reach the rank that would be expected of them without mom present. I found that overall, yes, orphans typically reach the rank of the rest of their family, even without mom alive in the group. However, orphans were slightly less likely to reach their family's rank than those who had a mother present in the group, so mom appears to play a role in some cases. Furthermore, among the orphans, the number of individuals an orphan had, the age she was orphaned at, or the rank of her living relatives did not significantly change her likelihood of reaching her family's rank. So that sort of gives further support that coalitionary partners may not be as important in blue monkeys as they are in the other species where rank acquisition has been studied. So to sum things up, we've found evidence that to reach their mother's rating, blue monkeys may behave according to their mom's rank, starting from a very young age. And they also may become more aggressive with age to help them increase their social rating. However, unlike the other species where social rank acquisition has been studied, did not find any evidence that with age they increasingly target group mates who rank below their mother and who they'd be expected to outrank in adulthood, and did not find any evidence that the number of coalitionary partners available matters to them either. So I'll leave it at that, but if anybody has any questions, I'd be happy to take them in the Q&A sessions after the presentations.
I see lots of clapping. Wonderful. Thank you, Rachel. Thank you also for prompting me. We will be setting up a series of individual Zoom calls immediately after this call for folks that want to ask questions of our speakers and congratulate them one-on-one. And we'll send the link for that momentarily and also at the end of the call in the group chat for everyone. So we will give you instructions about that at the end of the call, but there's going to be lots of opportunity for more in-depth conversations about the students' work afterwards. So our next speaker is Hannah Dancy. Hannah has worked on behavioral responses to olfactory stimuli of captive male tigers at the Bronx Zoo in New York. Her committee members were Pat Thomas at the Wildlife Conservation Society, Professor Marina Cords in our department, and myself. And her peers have described her as knowledgeable, passionate, creative, and someone who is probably going to use her black belt to take down Joe Exotic. So Hannah, take it away.
Thank you so much, Dr. Cross. Hi, everyone. Thank you so much. My research was behavioral responses to olfactory stimuli of captive male tigers at the Bronx Zoo. In case you don't know what olfactory is, it's basically anything you can smell, just scent. So a little bit of background, tigers are very obviously large carnivores, and they often have vast territories, up to 400 kilometers squared. They communicate over these large territories with a series of scent behaviors, and I'll show you actually in this video. So these scent behaviors are often very potent and long-lasting, and it can include something like spraying or scent scraping. So here's a spray right here where a tiger basically sprays a little bit of urine or urine fluid onto a tree, and this can last over a very long time. So in captivity, on the other hand, we have another issue I'd like to discuss. It's difficult to provide novel enrichment for tigers overall. Often a lot of items that we would provide to basically promote these natural behaviors are some sort of tactile item with a scent-based component. So a good example would be something like snakeskin or a large branch that was previously in a monkey's enclosure, and that would provide something for the animal to grab or claw and then also smell as well. So why are we looking at scent behavior? So different scents in recent years, basically scientists have been studying scent communication and have found that certain scents can change the behavior of wild carnivores, including coyotes, bobcats. And this is actually very important because it can provide deterrence for these animals in human landscapes. And that would actually lessen the sort of human-wildlife conflict that happened along these edges. And what we can also do is we can collect deposited scents, such as the spray like I described earlier, or feces, poop, and actually use that in genetic population management. We can take the DNA from those different samples. So it would actually be interesting to know what sort of scent behaviors we see in order to track these tigers and then be able to use this DNA in terms of population management and conservation. Finally, as I said earlier, enrichment in captivity promotes these natural behaviors, which is thought to then reduce stereotypic behaviors like pacing we see. And that's thought to increase the overall welfare of the animal. So here's my experiment. I decided to look at if different stimuli cause behavioral differences in tigers. So I compared 3 different stimuli. I looked at the urine of a familiar tiger, the urine of an unfamiliar tiger, and the urine of an unknown felid or a lion. So in order to define what a familiar versus an unfamiliar tiger is, a familiar tiger is a tiger that the individual has smelled, seen, or heard. And an unfamiliar tiger, basically the animal has not come into contact with them whatsoever. So here are a couple of my results. What I did was I looked at the scent events over the course of 90-minute trials. And I found that there was no difference significantly between the distilled water control I used and any of the scent stimuli that I used in this experiment. What I did find, what was interesting though, is that scent events are found to significantly decline over time. So when a tiger is first introduced into their enclosure, they will kind of go nuts. You know, they'll be sniffing all over the place. They'll be depositing different sprays. They'll be scraping the ground with their hind legs. And as they kind of adjust to their enclosure, this will kind of taper off over time. What I also found was that there was an increased frequency of scent events in lower temperatures. And this was most likely due to a reason I will explain on the next slide. So I had a couple of logistical constraints. And that was because I had difficulty acquiring unfamiliar tiger urine for my study. And because of that, a couple of my trials for unfamiliar tiger urine had to be performed at lower temperatures later in the season. And the cats that I looked at were, in this case, Amur tigers. And Amur tigers are acclimated to colder temperatures, which meant that they might have been more active during these colder climates, colder temperatures, excuse me. Another issue I had was a very small sample size, which is common in when you're looking at any sort of large carnivore. So a lot of my study was due to inter-individual variation. A lot of my data was kind of impacted by that. Overall, I would not recommend this for use in wild or captive settings unless more research is done. However, as I mentioned before, this is still an important topic of research. It's important in overall wildlife conservation, and it's important in order to provide good enrichment for our tigers that we see up at the Bronx Zoo. Thank you to everyone I listed here. And if you have any questions, please ask me in the Q&A session. If you are interested in talking to me about Tiger King, please go ahead, but be prepared for a lot of comments. Thank you.
Nicely done, Hannah. Okay, Maria Duigwasser, if you want to take it away to introduce Laura, I think, yeah, there you are. Oh, Maria, we're not hearing you, but I can see your face, your mouth moving. Huh, no. No, this is the second, Zoom is having crazy times today. This is the second call that this has happened on. I will introduce Laura. So Laura Plimpton is going to talk to us about the diet of urban cat colonies and the implications for zoonotic disease. Her committee members are Professor Maria Duigwasser, who is there and has audio problems, myself, and Jason Munchie-South from Fordham University. Laura's peers have described her as passionate, driven, full of surprises, eager to take on challenges, and the world's best cat mom, but also in danger of adopting every animal at the SPCA. So, Laura, go right ahead.
All right, thanks for that. So my project was on the diet of urban cat colonies and implications for zoonotic diseases. So in urban environments, cats form a unique social structure called a cat colony around a provided food source and are often managed through TNR or trap, neuter, return programs to reduce their population through spay-neuter. And so this results in a high density of cats in a given urban area, and with the need for food and mating mostly removed, also a significantly reduced home range for these cats. And so together, this creates a high concentration of cats in a given area, which while might reduce their impact at much larger spatial scales, which is common with true feral cats, may create a much more pronounced local effect on the urban community. And so this is an image of an urban feral cat colony. So here you can see 5 cats aggregated around a communal food source, and in this case, they're also provided housing for. And so a high density of cats in an urban environment may also have implications for the dynamics of certain urban associated zoonoses. So some common zoonoses associated with cats include rabies, cat scratch fever, toxoplasmosis, avian flu, Chagas disease, leptospirosis, and as of recent, COVID-19. And so while cats may act as hosts for some of these pathogens, they may also act as predators of common zoonotic disease hosts, namely rodents. So the urban environment may change the composition of available prey for the cats, which potentially changes the pathogens that cats are exposed to through predation, as well as the frequency of their exposure. And sort of the classic example of this is toxoplasmosis, where cats become infected by ingesting infected prey, like rodents, and then shed the pathogen in their fecal matter, like this cat is doing over here, which then contaminates the environment, subsequently infecting more intermediate hosts and completing the life cycle of the pathogen. And so my hypothesis was that the composition of a feral cat's diet is determined by prey availability, which is driven by habitat suitability for the prey. And this may alter the transmission dynamics of tropically mediated pathogens, as a more urban environment will have a different prey composition and subsequently different host and pathogen composition than a more rural environment. So to look at this, I collected fecal samples from 5 different cat colonies across Staten Island, and I extracted the DNA from all those samples and then amplified vertebrate-specific DNA and sequenced it on the Illumina MySeq platform, and ran a number of analyses to look at the composition of each of those samples and understand what the cats were eating. And so in this figure on the left, this is showing the frequency of occurrence of different prey items in the diet of those 5 cat colonies. And so what I found was that while cats rely primarily on cat food, most will also subsidize their diet with natural prey. Rodents were also the most frequent prey type, which were followed by birds, specifically Columbidae species, which are most likely pigeons in this case. And then most samples that did not contain cat food did contain DNA from natural prey, and those samples also had a higher frequency of DNA from more than one prey species. So next, I wanted to see how the diet of each colony compared to understand if there were certain features of the colonies that were driving differences among the diet. And so the figure on the left is showing the frequency of occurrence of each diet item broken down by colony, which are labeled A through E. And so when I looked at just the frequency of occurrence of the birds, which are in this box here, across all 5 colonies, I saw a pattern emerge where a high frequency of pigeons were associated with a low frequency of all the other bird species and vice versa. So I also saw this pattern emerge among the 2 mouse species, where a high frequency of occurrence of house mouse was paired with a low relative frequency of occurrence of white-footed mouse, and vice versa. And so next I grouped the prey items by urban adapted species, including pigeons, starling, and house mice, and forest adapted species, which included all of the rest, and again found an interesting pattern where the frequency of occurrence of one was negatively associated with the frequency of the other. So if we pull out just the 3 colonies where urban adapted species dominate in the diet, which are these 3 that are boxed out, and we look at them, we see that that high frequency of occurrence of urban species is paired with a low frequency of occurrence of forest adapted species. And then if we look at the land cover composition of these colonies, which is represented by the pie charts on the right, we also see that these colonies have more developed land overall, which is shown by that gradient of brown to yellow. And of that developed land, it is also more intensely developed, which is shown by this darker color browns and oranges. And so if we then look at the 2 colonies where forest adapted prey dominated, we see a similar trend again, where if there is a high frequency of occurrence of forest adapted prey, there's a low frequency of occurrence of urban adapted prey. And the land cover around these colonies was not only less developed overall, but if it was developed, was less intensely developed than the land around the colonies where urban prey dominated. So this led to my conclusion that forest adapted species are more frequent in the diet of colonies located in less heavily developed areas. And so in conclusion, the diet composition of these cats may vary even among colonies that would all be considered urban due to small scale variations in community compositions at each of the colonies. And that may have important implications for the transmission dynamics of tropically mediated pathogens within urban landscapes. And finally, I just want to thank everyone who helped make this project possible and who put up with me talking about cat poop for many, many hours.
Nice job, Laura. So we put the 2 kind of smelliest talks in a row, and I also, by mistake, put all 3 of our felid talks in a row. So our next speaker is Michael Levine. He's going to be talking to us about ocelots in the Brazilian Pantanal. Michael's committee were Professor Ruth DeFries, myself, and Allison Devlin from Panthera. And I should also mention that Michael came to Columbia to work with Professor Don Melnick, who passed away suddenly last year. And I think that we can all agree that Michael's excellent work, which you will see in a moment, is a testament to Don's legacy. Michael has been described by his peers as ecstatic, a friend to all, everyone's biggest cheerleader, the most enthusiastic person I know, fearless in pursuing his goals. So, Michael, go ahead and take it away for us.
That was very nice, peers. Thank you. I appreciate that. Hello, everyone. Thank you so much for coming. I'm really looking forward to telling you a bit more about my work, which focused on ocelots in the Brazilian Pantanal, which has got to be one of the most spectacular landscapes on the planet. It is a 140,000 square kilometer floodplain in the southwestern portion of Brazil. So that's an area about the size of the country of Nepal. And every year between the months of November and April, the wet season, it will flood almost in its entirety, leaving beautiful vistas like this one, but also underlying a really fantastic mosaic of biodiversity that's comprised of more than 3,000 species of plant and 1,000 species of animal. And right there alongside these fascinating natural systems is a really interesting human one as well. And it's centered almost entirely around the practice of ranching cattle. Cattle ranching has been a factor, both culturally and economically, in the Pantanal for more than 2 centuries now. And there's 95% of the land area in the Pantanal that's actually privately owned, and 80% of that is devoted to cattle ranching. Surprisingly, the relationship between ranching and the native Pantanal vegetation has been not especially antagonistic. More than 80% of the native vegetation of the region remains intact, which means there's some interesting lessons we can draw from this area about the coexistence of human and natural systems. It's not to say that everything's been peachy keen. There has been a fair amount of conflict, especially between ranchers and large carnivores like jaguar and puma, which has led to a wave of research in the last 10 to 15 years to develop sustainable and wildlife-friendly ranching strategies to reduce that level of conflict. And that's actually the backdrop for these 2 sites that you're looking at here, which were those chosen by my committee member and collaborator, Dr. Allison Devlin from Panthera. She deployed 2 camera trap surveys, one that you can see here in a complex of 2 different ranches, which hosted about 7,000 head of cattle at the time of her study. And this ranch actually ascribes to these wildlife-friendly practices, so they don't persecute any of the wildlife that they might find around the ranch. And she was comparing her camera trap study here to that of a park nearby, which was a complex of 2 national protected areas that completely exclude any and all human activity. And while she was looking at jaguar, I wanted to use the design that she set up and some of the bycatch data that she got. So not jaguar data, but ocelot data, to get a better sense of potentially how the human alteration of the landscape might be affecting the way ocelots occupy it. And I chose ocelots for 3 main reasons. They were readily available. They were seen quite frequently across both camera trap surveys. But most importantly, they're incredibly adaptable. And I thought it would be especially interesting to see if there were broad differences in occupancy between the ranch and the park, because if it's especially adaptable and there are still these broad differences, that means there might really be something going on here. And also, maybe really the most important is they are adorable. Look at those eyes. I just can't look away. So I had 2 main questions come out of this. The first one being, can this human alteration and use of the landscape, or haul as I termed it, be discerned using remote sensing methods, so satellite imagery, and then quantified so it can be compared across the 2 sites? And then second, maybe more importantly, does this haul signal affect ocelot occupancy of both of these sites? So to start things off, I did a supervised land cover classification in Google Earth Engine and broke things up into these 5 broad land cover classes to just get a good sense of what's going on the ground here. And as you can see, there are these large areas that very clearly look like human-altered spots within the landscape. And they seem to fall within these bare ground and grass and pasture land categories, capturing this large patch of pasture and also the roads that split off of it. So I simply reassign them as my haul signal, choosing those 2 as the basic components of it, and assigned everything else as a lack of haul signal. And when I ran my models, I used them as a covariate for ocelot occupancy in both of these places. So was it in fact a significant predictor of ocelot occupancy? No, not in the least. Neither was statistically significant, so not necessarily a super interesting story to be told there. However, there is another question that's raised by just looking at the overall occupancy figure itself. So on the right here in the blue, you'll see the occupancy percent of the ranch area, and over here in the pink, you'll see the occupancy percent for the park area. And it is a statistically significant difference, but also just a straight up substantial one. It's more than 30% higher occupancy in the ranch than there is in the park. And when it got down to thinking about what might be driving this, the wildlife-friendly ranching really came to mind. And I think it's that combined with the ocelot's adaptability that might be driving this. So there's no active persecution of the ocelots, which is great, fantastic. But on top of that, there's also been 200 years of ranching history that these ocelots have had time to adapt to. So they're relatively familiar with life on the range now. And I'm wondering if that's playing a role here. And the combination of the two potentially indicates that wildlife-friendly ranches could represent ocelot habitat in the ponds and all that is as good, if not better than protected areas, which has some pretty interesting implications for the long-term persistence of the species and its coexistence alongside this major economic and cultural force in the region. I want to express sincere appreciation to my committee, especially Professors DeFries and Cross, both of whom were absolutely invaluable over the course of this process, and Dr. Devlin as well. A special appreciation to Professor Melnick, as Professor Cross described earlier, my Quarren cohort members who kept me sane for the last 2 years, everyone in the admin office who made sure this all moved smoothly, and the rest of E3B for making this really feel like a home and a family, and I really appreciate that. So while I didn't necessarily win the ocelottery with my initial hypotheses, I do still feel like we got something good out of it, and I'm looking forward to talking about it in the session later on. Thank you all so much. I really appreciate it.
Thank you, Michael. Okay, I'm going to pass the baton over to Professor Mengi to introduce our next speaker.
All right, thanks, Sarah. So my name is Duncan Mengi. I'm on faculty in E3B. It was a great pleasure to have our next speaker, Alexa Helms, in my laboratory for the last 2 years. She was an integral part of the lab. Also on her committee, Matt Palmer, Bill Schuster, and Brendan Buckley. She, I think, is the first in the lineup so far to work on plants and bacteria. So she is diverging from the cat trend we had so far. She worked on a special type of tree called black locust deer that teams up with symbiotic bacteria that can fix nitrogen, which means they can take the nitrogen that's in the air and make it into protein, which is pretty cool. She specifically used tree cores to study how much nitrogen these plants are fixing and how they affect their neighbors, which is a really cool technique that she'll tell you more about. Her peers said that she is creative, which is an understatement. She's imaginative. She has so many talents. They didn't say this, but I'll add that along with Hannah, the two of them won the baking contest for our E3B Winter Games, I think 2 years running. So lots of talents. She's also an incredible artist, social planner, has the power to make me think nitrogen is exciting. She is the youngest but the wisest, according to her peers. So without further ado, Alexa Helms.
Sorry, it wouldn't let me unmute there. So thank you for that lovely introduction. So as mentioned, my thesis focused on how black locust trees influence forest growth, and black locusts as nitrogen fixers have bacteria that live in their root systems which convert this unavailable atmospheric nitrogen into more plant-filled versions like ammonia. And because black locusts, the nitrogen fixers in total, are relatively rare and that nitrogen is a limiting nutrient for most plants, it's really important that we understand the dynamics and the role they play in forest growth. And currently we have two sort of effects that we believe nitrogen fixers have on forest growth. The first is the fertilization effect, where as leaves from nitrogen fixers fall and decay, they fertilize the soil with fixed nitrogen, which is then taken up by non-fixing trees and facilitates the growth of these trees. Now, there are a couple of things that cloud our understanding about this effect, and one is specifically for black locusts. We don't know the rate at which they fix nitrogen or if it changes throughout their lifespan, which of course affects how much fertilization is actually happening. Nor do we know how long the fertilization effect actually takes place in order for facilitation of growth to occur. The second effect we consider is the competitive effect, which states that because nitrogen fixers are often early successional species, they tend to have higher growth rates than non-fixing trees and may become canopy dominant earlier, which allows them to outcompete non-fixers for important resources such as light, and this stunts the growth of the non-fixing trees. And much like with the other effect, we're not really sure how long it takes for this effect to take place and for an inhibition of growth to actually occur. So with that in mind, we decided to look into how the nitrogen fixation rates of black locusts are changing throughout individual trees' lifespan, and how forest growth overall is affected by crowding from black locust trees both currently and throughout time. And we were able to answer this sort of temporal element by looking at tree forests. And by looking at the tree rings, we came up with the growth rates of both black locust trees and neighboring trees at 2 sites in New York. And we were able to come up with the isotopic nitrogen values of the tree rings, which allowed us to infer the nitrogen fixation rates. So to answer that first question of how nitrogen fixation rates are changing, we have on our y-axis percent NDFA, which is percent nitrogen derived from fixation, and our yellow, orange, and red lines are the individual nitrogen fixers we measured. And you'll notice that these trees fix about 50% of their nitrogen, and that stays relatively stable throughout their lifetime. And you'll see, while on figure C, that it seems to decline slightly. That decline isn't biologically significant. So the big takeaway is that these trees are fixing large amounts of nitrogen at a relatively steady amount throughout their lifespan, indicating that a fertilization is actually happening. And in terms of how growth is being affected, we modeled the relative growth rate on the y-axis against the proportion of crowding from nitrogen fixers. And what that means is a value of 0 says that 0% of the neighboring trees of a focal tree that you're focusing on are nitrogen fixers, and a value of 1 means that 100% of the neighbors are nitrogen fixers. So if we look at the black line in the middle, that's comparing all of our trees together, and you'll see that as crowding for nitrogen fixers increases, so does the relative growth rate, indicating that nitrogen fixers are playing a role in the facilitation of growth. The next step we took was to compare non-fixing trees and nitrogen-fixing trees separately. So with the non-fixing trees, that blue line at the top, you'll see that this also has a positive slope, once again indicating that there is a facilitation of growth occurring. And as we know that they're fixing large amounts of nitrogen, and we see that nitrogen fixers are having a role in growth rate, this tells us that fertilization is likely the mechanism by which this positive growth is occurring. What's interesting is you'll see when we considered nitrogen fixers exclusively in that red line at the bottom that has a slight decline. And that's likely because the process of nitrogen fixation itself has a very high energy cost, and that is what is resulting in that sort of decline in growth rate. But I think what is most interesting and exciting about the research we were able to do is we could consider the past effect of nitrogen fixers on current growth of trees. So this graph is showing you the effect nitrogen fixers have in total versus how many years in the past we modeled, and there is a maximum of 14 years in the past at which this effect occurs. So what that means is that when a non-fixer is a neighbor to a nitrogen fixer that has been present and fertilizing the soil for about 14 years, that's when it will reach its peak in growth from the nitrogen fixer. So in conclusion, we were able to consider the sort of temporal element that had not yet been able to be addressed in this literature and research on nitrogen fixation, and we were able to confirm that black locusts have a positive influence on growth. And I was just really excited to be able to do this research, and I am so happy that I could achieve the honor of getting my master's with all of the wonderful members of my cohort, and I want to thank everybody who was able to help me along the way, and I'll be happy to answer any questions you have during the breakout session. Thank you.
Nice job, Lex. Okay, our next speaker is Emily Weisenberger. Emily is Emily started a really cool project looking at the effects of coyote presence on deer behavior, and she designed a perfect study, and then the bureaucratic processes at both the state and the city level like bashed heads and basically wouldn't let her do the project that she had put together. So she is in the midst of analyzing data on a very cool second project that she then had to design and start over again on. Emily is working on, as you can see, analysis of urban park occupancy by wildlife in New York City. Her committee are Robert Rockwell, or Rocky Rockwell, at the Museum of Natural History, Chris Nagy at the Gotham Coyote Project, and Marina Cords from our department. Her peers describe her as being organized, resilient, snarky, pushing forward through constant change to build a fantastic project, and that she will destroy you in Disney trivia. So go ahead, Emily.
All right, thank you very much, and I appreciate that. Snarky and trivia. So, like Professor Cross said, my project is on occupancy of species in urban parks within New York City. And New York City is a primary place to study this because of the recent introduction of an apex predator, which is the coyote, which was first spotted in New York City in 1994. And at this time, there are about 50 to 60 individuals that live throughout the city, preying on various mammals that live in these parks, such as white-tailed deer, rabbits, and raccoons. However, the coyotes are not found in every park, so there is a difference in these community structures already. This map here is just showing you a result of a camera trap study conducted in 2016. The yellow crosses here are where coyotes were seen and captured on camera, and you can see that they're primarily in the Bronx and in Queens. So since I've mentioned there are many different mammals that the coyotes are preying on throughout the city, New York City has the most amount of percentage of parkland of any city, with 14% of the area being classified as parkland, and these parks have a variation of different mammals that live there, including the coyotes, raccoons, squirrels, skunks, etc. And because of this, I'll be looking at 8 different species that are found in these parks to look at whether the community structure changes. So amongst them will be coyotes, raccoons, squirrels, striped skunks, white-tailed deer, possums, rabbits, and feral cats. And these are the species that are captured most often on camera traps. Additionally, things that will influence how the community structure is set up and how the occupancy can potentially change is where the camera traps may be placed, such as where they're in relation to a road, to water, the human density within 1,000 meters around the camera site, the canopy cover percentage, and the park size, along with whether coyotes are present or absent. So my main questions are whether coyotes will influence the occupancy of prey species, such as the deer, rabbits, raccoons, as I mentioned earlier, and what site covariates will influence it, and what combinations of these, such as will the distance to road, distance to water, and human density combined influence the species occupancy more than if they were looked at separately? Will the park size influence species that have larger home ranges, such as the white-tailed deer and coyotes? And will canopy cover influence species that rely heavily on canopy, such as raccoons and possums? So the data that I'm looking at is 600,000 photos obtained from the Gotham Coyote Project that were collected over 4 years. The Gotham Coyote Project studies coyotes within the city, so these camera traps were baited specifically for coyotes and placed in areas that coyotes were already known to be living, such as the Bronx and Queens that I mentioned earlier, or places where they could potentially be living or move into. This map here is just showing you the sites of where the camera traps were located. You can see primarily most of them are again in the Bronx up here and in Queens. And the sites that I ended up using were 51 sites in 26 parks throughout the city. So there are a few ways to go about this occupancy. The first could be using a multi-species model, which would take into effect the interaction of whether the presence of one species will influence the occupancy of another species. And this would be interesting to look at because I'd be looking at 8 different species and how this could influence each other. I could also do a multi-season, multi-species occupancy model. So this would allow me to look at all of the data over the many seasons and see if there are changes of occupancy throughout these seasons, as opposed to doing it by one single season at a time. These models here are just showing you the different setup that I will be using despite which direction I go. So I'll be looking at, as I mentioned, each covariate separately and then combinations of these covariates to determine whether they are more influential in how the occupancy changes. So predicted results based on the data that I've had and previous work that I've been looking into show that certain covariates may be more influential than others, such as the human density within 1,000 meters shows a decrease for all the occupancy for all the species. You can see here in this graph here, but the canopy percentage does not seem to have much of an influence on the occupancy, despite the species that rely heavily on it. So some preliminary results are showing that certain species, specifically cats, skunks, and squirrels, were found at every site, regardless of the covariates changing. So they had 100% occupancy. So those ones are not as influenced by these covariates as we first thought. But other species, specifically coyotes, which were found at the least amount of sites, at 41 sites, may be influenced by these covariates or combinations more so. And moving forward, I will be looking into how these combinations of covariates may influence these species and potentially see how it influences them throughout time. So just like to acknowledge and thank everyone on my committee and everyone who has been helping me thus far and will continue helping me as I move forward. Thank you.
Thank you, Emily. Exciting work. I don't know about you guys, how many of you live, or I'm sure many of you spent time in New York City, but I had no idea there were so many skunks in New York before learning about this.
I trapped several just last month.
Lucky you.
And a squirrel and a raccoon, 2 raccoons.
They're taking over. Nature is returning. So our last speaker this afternoon is Jared Meek. Jared is working on the comparative phylogeography to inform conservation of endemic pedicularis. I knew how to pronounce all of these things before I started, in the Hengduan Mountains of China. His committee are Darren Eaton, Ruth DeFries, and Richard Ree from the Field Museum at the University of Chicago. His peers describe him as wise, hardworking, calming, always willing to drop what he's doing and help, so genuine in everything that he does, our cohort's compass, and obviously has the best wardrobe. So Jared, I can't remember what Thursday is, but take it away. It's not Wildlife Wednesday.
It's Thrifty Thursday.
Thrifty Thursday.
Very kind of thanks, everyone. Yeah, so my master's thesis focused on the evolution of pedicularis, which is a genus of flowering plants, many of which grow in this mountain range in southwest China in Tibet, called the Hengduan Mountains. And here are a couple of these species right here. I think they're really wonderful and exciting to look at and study. So I've long been interested in why mountain ranges have such high species richness. This is a good map visualization showing high species richness associated with different mountain ranges across the world. So down in South America in the Andes, in Eastern African Rift System, and then where we focus our research in the Himalayas and Hengduan Mountains, you can see there's many more species than the lowland areas surrounding them. One of the causes of this that a lot of people have focused on is the process of uplift. So as mountain ranges form, they produce a wide variety of new niches for populations to move into and differentiate themselves based on different climates and elevations. So the Hengduan Mountains are here in the eastern edge of the Qinghai Tibetan Plateau in the Himalayas. They're significantly younger than either of these two formations, but they interact a lot and share some species. So we wanted to zoom in within the Hengduan Mountains and figure out exactly, if we could figure out specific barriers that were causing different species of pedicularis to differentiate over time. So here are the 6 species we focused on. I chose to focus on these because they're all very widespread across the entire sampling region. So this is a evolutionary tree on the left showing the relationships between these 6 species. And these colors correspond with the colors on the map, just showing where different individuals of each species were collected over the past 2 years. So yeah, over the past 2 summers, we went to the Hongguan Mountains, drove and hiked around to collect plants, pressed them for preservation, and also took leaf tissue and dried it in silicate gel so we could later extract DNA in the lab. And you can still see that as we were doing this, we were still able to enjoy some comforts of Wimbledon and the Women's World Cup as well. So we brought these leaf tissues back to the lab and extracted DNA. And then I used a specific type of genetic sequencing preparation called RADseq, and then sent those prepared libraries to get sequenced so we could get the genetic information of each of these individuals that we collected. So here I'm just focusing on showing you one species. This is Pedicularis rhinanthoides. And based on the genetic information that we got back, I was able to produce these maximum likelihood trees showing the relationships and genetic similarity between different individuals that we collected. And so each clade here, basically, in my mind, I thought would represent a different population, because if they're more genetically similar, they're probably closer together, sharing genes, which means they're clustering in different populations. And I validated this population assignment with these other tools that also use genetic similarity to group in different individuals into different populations. And once we had done that, I mapped these different individuals onto the landscape. So again, I'm just showing Pedicularis rhinanthoides, but across all 6 species, the main pattern that showed up was that these 4 river tributaries, these are all tributaries to the Yangtze River, but these 4 rivers were causing the populations to be segregated across the landscape. And then once we'd figured out that these 4 rivers were the main geographic barriers, we also wanted to see at what point in time were these populations diverging from each other. So I used another tool that estimates divergence time between different populations. So here the y-axis is divergence time in millions of years, and the x-axis is just the different species that we focused on. And you can see here the standard error bars are showing there's a lot of overlap between the divergence times of at least 5 of these different species, which indicates that multiple species crossed this gap, crossed this river barrier, and were split by this river at similar times in recent history, so between 0.1 and 0.4 million years ago. And that's important going forward because we can start to think about how these populations are being affected now as humans move into this landscape more heavily. And so here is that same Yajiang River, and across this landscape there are a lot of bridges and tunnels going through these different mountains that are connecting this landscape. And so we'll be able to use our information about what barriers actually exist or have historically existed to figure out how the genetic diversity of these different populations is being affected today. And I just want to thank my advisor, Darren Eaton, and the whole Eaton Lab, and the E3B department, and of course my fellow master's students. And it's been a wonderful time with everybody, and I've really appreciated all that we've learned from each other and together. And just big thanks to everyone who helped in the process.
Okay, well, that was super speedy presentations by all 7 of our graduates. And so I am going to send to all of you now, you should all have just seen a little chat bubble pop up, and that has a link to a Google spreadsheet, which has the names of each of our students, a link to a separate Zoom call. And for some of them, or many of them, you may have to enter a password, and some of them are those horrible automatically generated ones. So you just, once you open that sheet, don't close it, just keep it in a browser. If you do want to speak to a couple of our students, they will stay in their breakout rooms for about half an hour. So if you want to ask a few different students questions, hopefully you can kind of jump from Zoom to Zoom. But that is it for us on this large call. So what I'm going to do is unmute everyone. I'm warning you all now so that we can just give a round of applause to our graduates. So, over to you guys. Okay, well done, team. So go right ahead and go to those rooms and ask them challenging questions. about everything from Tiger King to evolution across mountain ranges. And I really want to thank all of you for making the time to be on this call with us today. And I wish we were doing it in person and we were sharing champagne and other goodies, but we'll have to make up for it and have an in-person shindig sometime, hopefully in the near future. So I will sign off now. I actually won't sign off because I'll keep this call open for as long as it takes everyone to leave it and get into those other Zoom calls. But if you have any questions about the logistics of doing that, you can feel free to stay on this call and Mari and I can answer them. But from us here at E3B, thank you all and congratulations to our graduates. Bye, everybody.